can start. Okay. So today is the last lecture for the first topic, which is the overview of the genome structure. Next lecture we'll get into sequencing and all that. So before I actually get started on this lecture, what we'll do is go over the writing instructions. So I read all the papers quickly. I was going to do it like piece by piece, but I think you guys can write proper English, so I'll just focus on the observations that I did while looking at those. So before I told you to write 100 to 200 words, that was just for the first one. From now on, it has to be about 400 to 500 words, so it gives you enough space to write. And anyways, if you're using different tools, then it should be easy. In terms of references, quotes, and paraphrasing, I see that most of you don't use references, so start by doing that. Last name, first letter, first name, comma, the year, or something along those lines, because anyways, if you're doing research, depending on the journal you'll publish in, they will tell you a specific format. So just show me that you understand one of them, okay? That's the first. Second, quotes. Quotes, we have to avoid using them because this is not an English class, and so when you're quoting, it's not telling me if you understand or not. You'll very rarely see in actually scientifically written papers published online that they use quotes. What they use is paraphrasing, and the reason for that is that you need to be able to argue while you're paraphrasing, okay? So you just don't go like, oh, the author wrote this, and then you talk, right? So you'd say exactly what he's saying in different words, and then continue the argument from there, okay? In English, in like an English essay, you would do that. But in this case here, you need to understand the science, and to understand the science is not to quote it, but to actually talk about it, okay? So that's that. I've observed two types of papers. One that is like 2-3 pages long, so the students are like going like deep. I don't mind those, but try to be concise, okay? Because what happens is that if you just write this much about a small topic, you'll just repeat yourself, okay? And you have a lot of words in there that have no meaning, right? This is easier to do for native speakers. If you're not a native speaker, you'll tend to add more words than needed. That's okay, but try to keep it concise. This is why we put limits on the wording, because you could write a freaking book about like one sentence, right? So it's like how much useless junk detail you want to put, right? Try to keep it in that frame. When you're writing a paper like this, you need to summarize kind of the article, so talk about the key points that are interesting for you, but that part needs to be the smallest part of your written paper. The biggest part should be your arguing, your thinking, your explanations, and your whatever you say is wrong or right. And this is where I need to see your thinking, because if I've seen some students, they would have, for example, 75 words of the 100 that are summary, and then 25 that are the opinion. That's not showing me that you're at this level in biology and that you're thinking. It's showing me that you're good at summarizing but not explaining your opinion, okay? So it should be the opposite. Try to usually do 1/3 summary and 2/3 exactly you, okay? This part. So 200 level classes, you would expect students to just spit out whatever they learn in class. Now, here it's 500, 600 for some people. Try to be original. 60% of the papers were about the CRISPR babies, and they were repeating pretty much the arguments that I was talking about. Some went a little bit further than that. So that's showing me that you're able to repeat what I told you, not what you think, okay? And so it's not something wrong. It's just now you guys are way too advanced to be just spitting what the professor is talking about. Like, bring some topic that is interesting to you and talk about it. Now, obviously, if there's one person that's interested in the CRISPR babies, makes sense, but 60% of the class, I don't think that's random, right? That's just people trying to, talk about things that are in the class. But now you need to, by yourself, explore different topics, okay? It's always good to make links to what we learn in class. So some people did this. This way it shows that you're able to connect what you learned with something outside. So this is obviously additional points for you. But again, try to have your own ideas, okay? So that's that. The next thing is discuss both sides of the argument, and although I tell you, like, tell me like the positive side, that means just tell me why you're against the other one. Why is this one the side that you're taking, right? And this is analogous to what we do in research. So usually you will have to write about your data, and some papers in there will be saying the opposite of your data set. You need to be able to say this is wrong because of that, and mine is right because of this. The thing that happens is that when you'll get evaluated by 3 peer reviewers and an editor, they know the field because they select them based on your area of expertise. So they know all the papers probably that are out in that field. So when they see something against another one, they will actually tell you to explain why and give you minor modifications, or if you destroy the paper, major modifications, but usually it's minor, okay? So try to do it right away from the beginning. So I don't agree with this because, and my point is valid because that, okay? Make sense? Questions about the writing? Oh yeah, and go easy on the ChatGPT. There's one person that has 85% of ChatGPT AI-generated text, because the AI can generate the text for you, but you can also train it to detect what's AI-generated and what's not AI-generated. It's not 100% perfect, but it's pretty close. And you can see it. I knew that paper was from ChatGPT. I just copy-pasted it, and it gave me 85%. And usually it has the form of like title, a little bit of text, title, a little bit of text, and instead of the asterisks, you put it in like, what do you call it? In bold, and it looks like it's written from you, but actually no. I don't mind. That's okay. You can do that. But I know for sure that you'll have problems with all this stuff here that I talked about, okay? Because ChatGPT does not necessarily do this on the go, okay? So try to at least filter it first and then go from there. Yes?
Do you have a preferred referencing style?
No. Choose one and go for it. Because I know that different papers will ask different styles, so I'm okay with whichever. Usually you'll use something like APA or Chicago or whatever, but Harvard, just use whatever you want. Okay, you can even put just in brackets the number and then reference it to like 1 is this, 2 is this, 3 is this, or put the names, whichever. Okay, all that works. Cool? Exactly, yeah. Instead of using quotes, so whenever you paraphrase something from paper, you have to put the reference from where it came. So that if I want to go check what you're saying, I can go and open that paper, right? Instead of just not knowing where it comes from, okay? Obviously, if you have only one paper you're working with, then clearly it's coming from there, but still do it. It's a good habit for you guys. Anybody planning to do PhD? No? That's it? I should have taken that decision too, but here we are. Okay, so we looked at how we go from blindly not knowing anything to finding pieces of DNA that have a certain effect on the phenotype of yeast. We then looked at the structure, so then how do we link both between the function and the structure, and this was done through the sequencing of the yeast genome. So this was done, it looks simple like this, but you'll see that it's actually pretty annoying and complex. So first, you have to define the structure of the genome, which we did. We looked at the different pieces that we identified through functional genomics, and then went on to understand their structure. When you have the structure, then you need to cut it into small pieces that could be sequenced, because the sequence is what we're interested in. Then you sequence everything, and then you have to stick everything back together in the right order, right orientation, etc., and avoid anything that is extra in your sequence, okay? So you do this for each of the chromosomes. There's 16 of them for Saccharomyces cerevisiae, so the yeast. One of them was sequenced by Concordia and Miguel. So chromosome 1 of yeast was sequenced by Concordia and Miguel, okay? Now, to do this, you need to understand maps. So this is my little tangent that that's an interesting fact that I found about maps, just to introduce the topic, totally unrelated, but it's quite interesting, okay? So this is the map that we all learned to use, right? Okay, the sizes here are super wrong. Does anyone know what's wrong? Yeah.
Africa is like
right, so not just Africa, but let me give you just an example. So you see this America here. Africa is supposed to be about 80% of America. So it should be from here to approximately here. Okay. I'll give you another example. Africa seems to be fitting 2 times in Russia, but it's actually the opposite. Russia fits 2 times in Africa, right? Algeria is supposed to fit 4 times in Canada, but I think it can fit like 20 times. If you take Algeria plus Brazil, it should be exactly Canada, right? So don't believe what you see out there. It's all bullshit. Actually, there's a lot of things that are pretty weird. So Russia, you can see it fits 1 time, and then you can do the other one here. It's about half of the African continent. Who knew this? Not bad, not bad. Okay, cool. So that's maps. Just interesting fact for you guys, okay? In terms of genomics or gene mapping, usually maps are used to position something relative to the other one. I know this seems to be clear, but it's not always. Some people would think that maybe it's actual distances, etc. In terms of genomics, when we did classical genetics, we were just trying to map genes relative to each other, not necessarily where they are exactly on the chromosome, okay? So there's 2 approaches to map the genome. The first one is classical mapping. Second one is molecular mapping, and both of them are used to get the yeast genome, okay? So we'll cover these 2. First, obviously, you start by classical mapping, and then you go to molecular mapping. So classical mapping, if you remember from genetics, well, probably very far, you have to follow the pattern of exchange of genes between the parents and the offsprings. So if 2 genes are very close to each other on the chromosome, they will go together most of the time. So they follow kind of this linked inheritance pattern. If they're far from each other, that means there's a possibility of crossover between them, and then sometimes they will not go together, sometimes they will go together, and that depends on a certain probability called the recombination frequency. Do you remember that? Kind of? Okay. So what we did before in classical mapping is that all we did was we were taking these genetic markers. So this is markers that you can see in the progeny, color or whatever, something that gives you a phenotype. And we tried to look at the offsprings from a parental cross to see if the, why is this moving always by itself? To see if those genes were being carried together, and then if the progeny had those 2 markers or just 1 or none or whatever, okay? This would tell us if they're on the same chromosome, different chromosomes close to each other, far from each other, and there were a lot of markers on the yeast chromosome, so it made it very easy, okay? Most of them were within the, close to the center of the chromosome, but some of them were established along the chromosome, okay? And for yeast, that looks like this. So yeast has 16 chromosomes. These are all here. Forget about the colors here. It's just a picture I used for the purpose of showing the chromosomes. But as I said, most markers were identified close to centromeres, and then there were others that were distributed along the centromere. So if, for example, geez, these guys. If you're trying to see if a gene is on the same chromosome or a different chromosome, then you would try to see if they are inherited together, separately, and how often. If they're on different chromosomes, you would technically expect them to be separately, right? To be more together, actually at equal frequency together. If they are on the same chromosome and very close to each other, then you would expect them to always be together, right? Make sense? So, do we go party with them, or what do we? This is very annoying, very, very annoying. Okay, let me just, anyways, so that doesn't, I have a little bit of attention. I'm always everywhere, okay, so things like this, they disturb me a little bit, but what was I saying? So this doesn't get us the sequence, okay? To get the sequence, we actually need to do a few things. We need to get the chromosome into pieces that are of an appropriate size to be sequenced, and we need to understand what this piece needs to look like, okay? So we'll be looking at this now, how you go from a full genome to actually the sequence itself. And this is basically the topic of today's lecture. So the first step you take your genome, break it into small pieces. Now, how big are these pieces? You have to figure out how big they are, what shape they are, etc., right? But before we get into that, we have to kind of figure out which method we'll be using to break the pieces, right? So you have to think of it as you being in 1999 and not, or 1995 and not knowing what the hell it looks like, right? So these questions, you have to answer them before actually. Now it's clear for us. You use restriction enzyme, put them together sequence, right? But at that time, it was not clear, right? So they were kind of going in, not blindly, but somewhat blindly, right? So there's two ways to do it. You can do physical shearing, or you can do restriction enzymes. Physical shearing is, for example, ultrasonication. So if you take a cable like this and you wave it like this very fast, it will break, right? Because of the tension. So ultrasonication, all it does is that it will send pulses, and the pulses will break it into small pieces. You can also take your genomic DNA that you extracted, pass it through a small pore, a porous membrane, and because the pores are very small, it will just bend it and break it. You can do a lot of things. You can even vortex it super fast, and then that will also break the fragments, okay? Now, the size of the fragments depends on the time and the intensity of the mechanical force that you're using. If you put a lot of force and you spend a lot of time, your fragments will be super small, right? If you do the opposite, they will be longer, okay? And they produce something called a blunt end. So it cuts directly in the middle of the two strands. It does not create these overhangs that we'll see. Cool? Simple? Okay. We still use physical shearing, but for other techniques, because they're important into getting to get equally sized fragments. Okay. Restriction enzymes, they are not like the physical shearing in the sense that the restriction enzyme sites where they cut are not equally distributed along the DNA. And so when you use a restriction enzyme to cut the DNA, you'll end up with different sized fragments. And the problem with this is then how do you find the right vector where you put these fragments? Some vectors can take up more DNA. Some, a small 2 kb vector will never take a 50 kb DNA fragment. Okay, so things like this. So these restriction enzymes, they recognize these sites, right? These repeat sites here, inverted repeat, and then all they do is that they cut at a specific position, either inside or outside, and they will create these overhangs, right? And these overhangs will be important because then you need the same overhang on the plasmid so that they can stick together. This is why it's called the sticky ends. Okay. Here I'm talking about 4-base site and a 6-base site. I'll give you an example right away. For yeast, they use these SAW3A restriction enzyme, which recognizes this site here, GATC, and cuts behind the G, right? This enzyme here is known to cut up to, it's approximately 250 nucleotides, the fragments that come out of it, okay? If you completely cut the genome of yeast, by completely, that means you allow for all the cut sites to be targeted by the enzyme, then you'll get about these fragments that are about 250 nucleotides on average. Now, this is not good. You need bigger fragments for cloning, okay? So then you have to figure out for how long do you let the reaction to happen so that you get a specific size of fragments, okay? And this is why what you do is that you don't go for a complete restriction enzyme cutting, but you go for partial digests. So you don't digest completely the genome. And to do this, you need to use different variations in time and amount of the enzyme, and then you need to do gels and see how big are your fragments, okay? And so in this case, for the SAW3A site, they are spaced at about 250 base pairs each, but not always, okay? And so for you to get these 10 kb fragments, which are typical of cloning, you would need to cut 1 out of every 40 sites. So that means you cut here and here, not in between, right? Now, how do you do this? This is, again, what I mean by time and the amount of the enzyme. So you would have to put different timings and see if there is a time that works where you have a lot of 10 kb fragments and not so much the 1 kb, 2 kb, 3 kb, etc. Okay? In the case where, this is just a theoretical example, in the case where the SAW3A site is at every 10 kb distance, then if you cut it to completion to get this 10 kb, then what's going to happen is that your partial cuts will be 400 kb. Does this make sense, how this goes? No? Maybe? Okay, cool. So the genomic library. So how did we do it for yeast, actually? Before I talk about that, we did not use blunt ends because, yes, they are of the same size, but they're just harder to clone. So instead, we use those with sticky ends. And for this, we have to optimize the shearing time. Okay. So the first step is to run those digests on an agarose gel. So let's say you cut them at a specific time and amount of enzyme. Then you will make multiple lanes on your gel. Some you have cut for more time or less time. And then you'll have one lane that you cut to completion and one lane that you cut approximately 1 to every 40 sites. And then you collect the fragments on your gel that are between 7.5 kb and 12.5 kb, and that should give you an average of 10 kb fragments. Obviously, some of them will be longer, some of them will be shorter, but you try to get that whole region. On a gel, it looks like this. So let's say you have your ladder here with a molecular marker that tells you what each line, what each position here is in terms of bases. So here there's 1.9 kilo bases, so 1,900 bases. Here there's 9.5, so we're looking at this range here, right? This is where we're trying to figure out our 10 kb fragments. A little bit above to get the 12.5 and a little bit below to get the 7.5. In the first lane, if you take a look at this gel, you see that here there's nothing. That means the DNA has not traveled in this region, so it's only stuck here. So here, your fragments are way too big. They're on average 23.7 kb. So then you have to look at the other lanes to find the lane where this middle line here is around 10 kb, okay? And then you have to cut this piece, extract the DNA from it, and you'll have fragments that are 7.5 kb, 8.5, 7.6, 7, 8, whatever, okay? Up to 12.5. Make sense? Easy? Okay. So as you can see, this is very annoying because it's a lot of work. Okay, now it's much more simpler, and we'll see that at the end plus next lecture. Cool. So we have our fragments. We decided that 10 kb is the right size. That should be easy to work with. We got our 10 kb fragments by doing these restriction enzyme digest and then getting them through the gel and extracting them from the gel. Now we need to build our genomic library. And the genomic library is basically putting those fragments into plasmids. For the SAW3A site, you need a different site on the plasmid so that they can bind to each other. Okay, so this is where you have the four units for the SAW3A. So this here, right? And then you need the other side. So the plasmid needs a overhang of 5 plus the this here. So it needs to be a site of six units. Okay, this is where the four bases cut, and then the 6 bases. Do you get this? Make sense? Okay, so the enzyme that cuts the plasmid, well, one of the enzymes that you could use is BAMH1. So BAMH1 is used for the plasmid, SAW3A is used for the genomic DNA from the yeast, and then you build all these different plasmids with all the different fragments, okay? Now I just want to go backwards here. 10 kb means what in this case? It could start from here to, let's say, here, or here to here, here to here, so you'll have sequences that overlap, right? So we'll see in a second how we can filter out the sequences that overlap so that we only get the exclusive parts, okay? Okay. In this case, we're using a plasmid to be replicated in the bacterium that you will transform to amplify our plasmids and have our library that we can store, okay? If you want to sequence it, you need a special plasmid because the result from the replication is not the same, right? If you remember Sanger sequencing, you need a single-stranded DNA so that you can add the primer and then add the nucleotides one by one to get the sequence. Do you remember that? We'll be taking a look at that next lecture, if I'm not wrong, or the one after, okay? So these plasmids that have this property are called phage mids. It's a mixture between bacteriophage and plasmids. And what's special about them is that they have two origins of replication. The first one is coming from the, this one is for the bacterium, the regular origin of replication that you have for bacterium to replicate this plasmid during the life cycle, cell cycle of the bacterium. The other one is a chimeric origin of replication that comes from the, in this case, is the F1 phage. So this is just a virus that infects bacteria. Sorry for that. I think there's an animation on these slides that moves the, I will take care of it for next lecture. So when this origin of replication is actually activated, what you're going to do is produce a single-stranded DNA molecule of the plasmid and not a double-stranded plasmid, okay? And then that will be interesting and important because then you can do Sanger sequencing on that piece. And if you know your DNA sequence for the plasmid, then you can do that minus the gene, and you'll know the gene basically by just removing the sequence from the plasmid and leaving just the additional stuff that you put there. You probably guys have seen P-Blue script. Well, it is a phage mid, okay? This plasmid was used a lot in the early days. Now, not so much. There's much better plasmids that were designed. Cool? Simple? Good. Okay. How do you know that your library is a good representation of the genome? So if you're cutting different pieces of DNA and then trying to put them into plasmids, it's not as simple as just, oh, I cut them. Now, if I put enough plasmids, I will have each sequence inside the plasmid. Some sequences will be incorporated more and some less, right? Because of their size, because of the bacterial response to them. And so you need a way to figure out how good is your library in terms of representation compared to the genome, okay? So if your genome has 10 genes and you make your library using a cut using this enzyme, it doesn't mean that your library will have 10 genes. It could have 7, 8, so you have to find a way to know how representative it is of your genome. This assumes that everything else was perfect, okay? So what I'm about to show you assumes that everything else is perfect. You did not make a mistake in the shearing, partial digestion, in adding the linkers, in the ligating to the vectors. So this is just like purely theoretical, okay? But it's a good idea when you're trying to do this kind of work, okay? So the amount of genomic DNA in the clones has to be multiple times the amount of DNA in the actual genome, okay? So if you have a genome that is, you have 1 nanogram, your genome is 1 nanogram, then usually you need to put 5 nanogram of that genome to have a good chance of having all the pieces into your vectors, okay? Because it's quite random, and some regions are more clonable than others, and we'll see why, okay? So this in research, it's called the genome coverage. So it's the ratio of the amount of DNA in your library compared to the amount of DNA in your genome. And usually it needs to be higher than 1 for your experiment to work, okay? So 1x means 1 genome coverage. There are other ways of calling this. You can call this the number of genome equivalents, so the number of times your genome is in there, or the genome redundancy. Jeez. So again, as I said, some regions will be more present than others, and so this is why you need a multiple of your genome. What do I mean by some regions will be more present than others? So you see this region here in the middle? This region here will be probably in 1, 2, 3, 4, 5 different fragments, while this region will be only in 2. Right? So this is why you need a higher genome coverage to ensure that this one here will be present in one of your vectors. This one for sure will be, because you have it 1, 2, 3, 4, 5. When I say this region, I'm talking about this up to here. Yep.
So if you're trying to increase your genome coverage, can you do it? Do you have to do it like when you're creating a library, or can you just amplify your library and then that is good enough?
No, at the beginning, before having the library. It's not like I'm adding 5 times the amount of DNA. No, yeah. You need to have it. That's a very good question. You need to have it at the beginning. Okay, so you would do different cuts and have it like that. Okay, let's do some simple calculations. Okay, so the way they did it, these scientists, is they looked at probabilities. Okay, and the probabilities follow a Poisson distribution. If you don't know what that is, you probably know the Gaussian distribution. So Poisson is a similar thing, just that it's tilted towards one side. Okay, approximately. Okay, you can read more on this or take it in the biostats class. But in this case, the Poisson distribution has a way of calculating things, and we'll take a look at it right now. Okay. So let me explain this. The probability to have 63.2% chance of getting one region, any given region, requires one genome coverage. So if you cut the DNA, the genomic DNA, once, and you just have one equivalent of the genome, and then you run it through this process, you don't have 100% chance of getting all the pieces. You have 63.2% chance of getting any given sequence. So there's basically 27% that is missing, because some regions are represented more in the fragments than others. For example, this one will be in more fragments than this one. And so technically, you have more chances of getting this than that, and this will be part of the 27% that I told you about. Okay. If you need to get very close to 100% chance, then you need 10 times the genome. Okay. And even then, this is like, again, it assumes that everything is perfect. Okay. In reality, you'll need more, and I'll tell you why you need more. Does this make sense? Okay, some calculations. So one of the only formulas that you'll see in this course, which is highly underrepresentative of genomics, it's actually the opposite. If you continue in this field, you'll do a lot of calculations. So let's take a look at this simple one. So the ability to predict the number of clones at a certain probability using a certain genome goes as follows. So it's n equals to the natural log of 1 minus p. P is the desired probability. Obviously, if you need 100%, then you'll include it here as 1, right? Divided by the natural log of 1 minus f. And 1 minus f just means how long are your fragments over the total length of the genome. So if we do that, and we read this example here, so to achieve 99.3% probability of having any given part of the genome into your vectors, into your library, then you need, let's say your library is of 10 kb fragments, and the yeast genome is 30 million. Then you'll see that if you do the calculation where you put your probability here as 0.993, and then the ratio, so f is basically this here, 10,000 base pairs over 30 million, then you'll get this number, right? So what does this number mean? This number means you'll need this many clones to get this much, right? This is a lot. I'll just give you an example from my project. Usually I do times 10, so if I need to take a look at, well, not times 10, times 100, so if I need to take a look at one CRISPR mutation, then I usually try to get 100 clones and then grow them, so single cells that I think had the CRISPR, and then 1 or 2 of them will have my actual mutation. Imagine you need all this here. This is for yeast. Now, for human cells, the number is like in the billions, and not even the billions, the trillions. Okay, so we needed a better way of doing this. Now, these probabilities are not perfect. They assume, remember these assumptions, okay? They're important. Also, this slide is an example of looking at the limitations of a technique. So this kind of thinking needs to be included in your writing. So the probabilities, so let's say, for example, you're reading a paper that I assigned to you and you see that there's limitations in that paper. Well, you have to think about these assumptions here that they're making, and do they make sense or not, and why, okay? In our case here, these probabilities that we are measuring, they assume that the genomic regions are equally clonable. And that's not true, because some of them are going to fit much better within that plasmid and others not. Plasmids tend to be finicky and annoying, so not all regions, just because of the molecular structure. Sometimes the insertion will be in the opposite side, which will kind of make you do mistakes. And also, this slide is very important. Okay. Also, this piece here will be in more plasmids again than others, so it is more clonable than this piece. Make sense? Because there's more sites surrounding it and more pieces than, let's say, these ones here. Okay, let's get back here. And then also, and this is because what happens is that there's two reasons for this. The first one, the restriction enzymes are not the sites are not equally distributed among the genome, and so in that case you'll have sites that are closer to others, so they will be flanked by multiple different fragments, while others will be super far, and so you'll have one fragment of that and not, right, covering that region. So that's that. Second, the basis of CRISPR. So CRISPR recognizes these palindromic repeats and then breaks them down. So bacteria have this immune system, kind of like a very primitive immune system. This is from where CRISPR came, that basically breaks this type of DNA. So if your DNA that you're trying to insert into the bacteria has, so the fragment has a lot of these repeats, the bacteria will kind of try to kick it out and break it down. Okay? So this is why some regions are more clonable than others. So how the hell do we fix this problem? Well, you put more material when you start than what's suggested by the Poisson distribution. So you can do 20 times more, 30 times more, etc. And obviously this is super annoying. It's a lot of work, but it works. Okay. Let me see what's happening here. Okay. So if you remember at the beginning, I was talking to you about classical mapping and then molecular mapping. So classical mapping is where you have, as I told you, you have these markers and then you combine them in a binary or ternary way so that you see which ones go together and which ones don't go together. Then you know if they're on the same chromosome, close to each other or not. When you have all these structures, then you need to figure out where a gene starts exactly and ends exactly and in what order they are. So for that, you need to create a physical map, right? And this is basically the physical map so that you know this gene starts from here to here, and then this region here is the same as here, right? And this one is the same as here. This is referring to, again, these fragments here, okay? Okay, so how do we do this? So before we get into how we do this, I just want to talk about the initial idea and now where we're pivoting. So the initial idea was that we're trying to understand how the genome is organized. So we cut it into functional parts, and then we find the smallest functional part, so the smallest piece of DNA that had a function. And then let's assume that region is 10 kb. The main region is 10 kb, then you need to cut it into smaller pieces, and then each one needs to be inserted inside the plasmid, the vector, and then tested for function. And then you would pinpoint in that 10 kb where is your gene. This is a lot of work, okay? It doesn't seem like it, but you can end up with thousands of 386 wall plates, okay? So that's a plate like this with 384 dots, and you need to fill each one with a reaction and then compare them, see if they have a function or not, okay? It's a lot of work, and there's not as much freezers. So the way you solve this, you just sequence the whole genome, okay? Very simple like that. So you cut them into small pieces, sequence each one of the small piece, and then try to assemble everything back together, okay? See how they are connected, because they will have overlapping parts. Okay, so first part, we need to get the chromosomes. So the first part, you start with the genome, you separate it into different chromosomes. For the yeast, this is the gel, there's 16 of them. As I told you, one of them was sequenced by McGill and Concordia. Obviously, it's chromosome number 1, so we're that cool. And there's other teams that did this. Actually, I'm actually curious. Let me just go see the size of chromosome 1 compared to the others. Okay, we took the smallest one. These guys. It's okay. At least we were part of the effort. So you would run this whole genome on a gel. The old pulse field electrophoresis. So this is a bit different from SDS and agarose and all that. You separate the chromosomes based on their size. Then you purify the DNA for a single chromosome. Obviously, we took the smallest one. We could have, up the level, but it's okay. And then you have to build these contigs, okay? So you take your chromosome, cut it into pieces using these restriction enzymes, and you'll have, for example, something like this, okay? As you can see, there's an overlap here. So these are called contiguous sequences. They are continuous, but they also overlap. This is why the word contiguous. And so you'll have to sequence not all of them, but some of them to be able to find out what is the actual DNA sequence, okay? There's no need to sequence this one if this here is sufficient, okay, for example. And so to avoid doing all this work and sequencing all the pieces, you will create these libraries of contigs first, so that you only take the ones that, the minimum number of sequences to sequence, okay? Because otherwise you'll never be done with this. So let's take an example here where you would take this sequence here, this fragment here. You would put two probes on it, on each end, and that will tell you when you do, for example, your gels and all that, and you use X-ray to figure out if a certain band has that fragment or not. So if you put, let's say, the red probe, you will see that two bands will appear. This one here, the original one, and then another sequence. And so you know that these ones are overlapping, right? Because you're using a probe from here, and it detects not only this, but this. If you use the yellow probe, you'll see that these two pop up, but not this one here, because the yellow probe is not overlapping with this, while this one here is. Obviously, if you put two probes, you'll see that this and this sequence appear, and so you know that these fragments have this plus other sequence, so they are overlapping. So then when you sequence it, if you sequence this one and this one, you know that this one here has this piece additional and this piece additional plus the overlapping section, okay? This avoids you sequencing all the fragments. For example, here, you would not need to sequence this one, just this one is sufficient, okay? Make sense? Now in the lab, how do you do this? Plates, a lot of plates. Yeah. If they are contigs or not, it's the way they react to the probes. If you see two bands that appear with the same probe, but they are different sizes. That means that those sequences are different fragments, but they overlap. Yeah, we'll get there. We'll get there. We're not there yet. It's Sanger sequencing, basically. So actually, we did talk about it a little bit. I'll get back. So these pieces here, you will put them in the vector, right? And then you'll activate the origin of replication from the phage, so the F1 origin of replication. That will give you a single strand of DNA. And then that's the one you sequence using Sanger sequencing. Make sense? Is found in both, and so it means they're overlapping. Make sense? So you can see how it's a lot of work, right? Crazy. Okay. Now, to avoid breaking the cells and making a million different plates, you will take all your digests with all your contig library. You will put them in vectors. You will put them inside the bacteria. You will spread it on a plate like this. And then you'll take these paper or nylon, whatever, these membranes that you will put on top of the plate. And that will carry a little bit of bacteria. You move it to the side. You keep a plate for you to just grow and have those colonies and keep moving them to new plates because the plate will fill. And the other one, what you'll do is you'll move it. You'll do replica plating on a new agar plate. You will denature. You'll basically fix the DNA and break everything so that then you can hybridize it with different probes. Okay? So you'll have your bacterial DNA that's fixed on the membrane after doing different washes and different detergents, whatever. Okay, you just put the paper inside these solutions, take it out, put it in the next one, take it out, and then you'll have this DNA on the paper. And that DNA is in the same pattern as your bacterial cells before. So then you'll take a probe and you'll run it against this membrane, and it will tell you which colonies react to that probe. And then you'll know which fragments you're dealing with. Okay? And that's the ones you sequence. Cool? This way you try to figure out the least amount possible of fragments that you can sequence to avoid sequencing all the colonies' DNA, and that's too much work. Okay? So instead, you do it probe by probe until you figure out the minimum amount of colonies you need, and those are the ones where you extract the DNA and go for sequencing. You redigest the, you run the F1 origin of replication, you get your single strand from those, and then you sequence it. Okay? What else? What else? Okay, so we have the sequence. Now, how do you put everything in order? One way to do it is to use restriction enzymes. So this is restriction enzyme mapping, where you would use multiple different restriction enzymes to cut these pieces. So let's say you have a contig, 3 contigs that overlap. You need to figure out how they go, right? The direction. And so for that, you need multiple restriction enzymes, and you need to cut at different spots. Okay. Again, with the same fragments, you can use 2 different, restriction enzymes, and take a look at the fragments that come out from those fragments. Okay. So here you can have, for example, H stands for HIN3, and then P is PST1. So these are 2 different restriction enzymes, additional to the SAW3 that was used at the beginning. You will cut them, run them on a gel, and then depending on the size of the resulting fragments, you will know which ones are which. So for example, in this case, you'll have, for this reaction, you'll have one band that is of this size. So you know that this band, this band, this band, and this band are the same. And then you have 2 additionals from here and from here. And then you compare them to all the others. This you should have done in genetics, so 261, where you're giving a gel with 4 different lanes, and then you have to figure out the position of the genes relative to each other. Okay. Obviously, in the exams, I'm not asking you to tell me to read the sequence and all that. You just need to understand the history and the science behind it, so that then you can talk about it, okay? And then you just do the same, okay? This is just showing that when you have those pieces, then you have to actually use these probes and then go from there. Same way, cut, probe, figure out the order, cut, probe, figure out the order, okay? What else? Limitations. So this is a lot of work and very old school, but this had to be done previously so that we can get this. So what you do is that usually you will collect your first round of data, then you'll generate, you'll identify the overlaps, you need to find new probes, and then you repeat. You cut again, find overlaps, define new probes, and then repeat the process again. Because what I didn't tell you is to build the probes, you need to actually just study this piece here to figure out what probe you will use for this piece, okay? Now imagine 12 million base pairs or 30 million base pairs. You need to study the pieces themselves to make probes to then figure out which ones overlap and which ones don't, okay? So it's a lot of work. This is why we do sequencing, okay? And every time your collection of clones will get bigger because you'll have more probes to find the overlapping sequences, and then you can keep those. Because this is sequential, you kind of need to do it in this order. So you find your first set of data, identify the overlaps, get the probes, repeat, and then you make your, what do you call it? You make your library bigger. When you're dealing with current methods, it's parallel reactions. So you can run multiple of these in one run, right? And so this is much more faster. Otherwise, we'd still be doing this. And that's a lot of work for human cells. Obviously, yeast is manageable. This was like 100 different scientists that worked on this. But for humans, it's too much work, okay? One more thing. When you have your 10 kb fragments, they're not sufficient to be sequenced. You need to cut them into smaller pieces that are sequenceable, okay? So you keep cutting until you get to the sequence. So 10 kb does not work. Sanger sequencing has a limit of 1 to 2 kb if you're perfectly doing it. Usually it's around 1 kb. In the lab, I'm only able to get 700 base pairs, so so it's not bad, but if it's for sure you cannot do the 10 kb in one run. You have to cut it and then further and then do that. And so obviously you will cut it, the 10 kb that you find, into 1 kb or 2 kb, put them again into sequencing vectors, and do all that, okay? So these are the limitations, because you still need to do some cutting. It's not parallel, it's sequential, and so obviously it takes a lot of time. Good, so there's a little but a little picture of what this actually means. So you have your pieces, digest them, have smaller pieces like this. PCR, we didn't look at it, but we'll take a look at it. You have to put these adapters so that you can, because you don't know the sequence, right? So let's say you have your fragment like this. You don't really know its sequence, so you need to attach an adapter to it, where then you would have a primer that responds to that adapter, and then you can go and sequence it, okay? So you attach those adapters, you multiply your DNA, and then you sequence. And then obviously you need some computational power to put all this back into place, okay? Did I forget something here? Forget this part here at the bottom for now. We'll get into it a bit later. Cool. Let's start taking some points for for participation. So now that you've seen this, and you know about the structural and all that stuff. By the way, don't fight to answer, it's okay. There will be enough time for everyone to get the points, and I'm not crazy about that. But what do you think are the ideal characteristics of a sequencing procedure? So you've seen the BS way of doing it, the very long and annoying and tedious way of doing it. What's the actual ideal way of doing it? I will get to you in 1 second, but these questions are very representative of exam stuff. So think about them, okay? So give me at least one or two or few characteristics that that are ideal. Start by you, and then you. Yeah.
I mean, you would want the longest read possible in one go.
Longest read possible. Well, yeah, that could work. How?
Like if you could read the whole genome in one go, you wouldn't need to do any digestion or anything.
Yeah, that's the perfect, perfect approach, right? Yeah, that could be an ideal sequencing procedure. Let me think if there's a problem with it. So then, does anyone see a problem with it? Yeah.
Accuracy. Accuracy could be like bigger the size of what you're sequencing, the less accurate it is. Especially if you have like homologous like poly A tails or anything like that.
Okay. So just 1 second. I'm getting to you. So when you're sequencing, this is true. So when you're doing a Sanger sequencing method, so we don't know this now, but I'll show it later. When you run the sequence, you usually don't trust the first 100 base pairs that you sequence because it's not precise. Because the primer attaches, sometimes not, and then the enzyme that takes care of doing the replication will not be fully attached, and so it will be like there will be mistakes in it. So usually you let it run a little bit until it attaches properly, and then you determine that. So you need to put it inside other sequences and then play with that, right? So that you would have kind of the first part that is garbage and then the right sequence. The end also is a problem because the enzyme will try to detach, so you'll have that. Yes? What's your name, by the way? Matthew. Okay.
So there are more modern techniques, like there's the Oxford Nanopore, the Pacific Biosciences method. They are both really highly automated, so there's almost no human component to it. So this is purely, you can just let it do its thing. It has very high throughput, which is another very sought after component.
Yeah.
And you can most of the time trust its accuracy because of the computational element. There's no room for human error as there was in the early Sanger sequencing, where they had to actually pay researchers to look at their base pairs one by one.
Yep. Did anyone understand this? So there's new techniques that exist which work on these small chips, these small little slides, and use microfluidics and things like this. So nanopore and all that, they have these adapters, they link the DNA to it, and that DNA is just shaking like this, and then you can just replicate it multiple times and sequence from there. They use a lot of probes that are emitting light and all that. And the light is detected by your spectral and then just, recorded as a sequence. So it avoids the mistakes that happen due to human error. For example, improper shearing or just improper analysis or things like this. So this doesn't make sense. So I guess the ideal sequencing procedure, do you have something to say?
When it's like high throughput and you use like shorter sequences, you would have like higher error rates compared to the Sanger sequencing.
If you have shorter ones? Yeah, exactly. So the idea would be
having like a long sequence wouldn't be
more error or less error.
than use like Sanger sequencing.
The
Low throughput would have like lower error rate.
Yeah. So the reason why there is a problem with the size of the sequenced fragments, it's because the enzyme that does the replication, will detach at some point. It will just get away. So you need a specific size. But the ideal procedure would be a one go without getting off, right? So if you were to take the full chromosome and then put the polymerase that will do the replication, which is part of the dideoxy Sanger sequencing, at some point, even though there's no reason for it to get out, it will get out. And usually it's about 1,000 to 2,000 base pairs. Okay, so ideal sequencing could be perfect enzyme that just goes through the whole thing. Something that's high throughput. So the high throughput, you would need to cut your DNA in smaller pieces and link it to a slide and run that slide in a machine. Do you have something, Arthur, right?
Yes, I know there's the iron through
Yeah.
And then you have to sequence that. But I think it's high throughput anyway than too, but
I
it's not accurate.
I don't remember. So it's a technique where you would have an ion flow.
Yeah, and each nucleotide modify the pH by a certain amount.
Yeah. Okay. Yeah. I think I saw this somewhere, but I don't think it's used. I don't. I've never heard of a company actually using that. So probably it was like used for a little bit and taken out. Now there's nanopore, as he said, and many others that are working very well. But the idea is high throughput, remove the human side to it, and then just kind of automate it in the sense of detecting the nucleotides. Obviously, there's probably strategies where you could use a little bit more chemistry because Sanger sequencing is really based on chemistry. So I can't think of any right now, but there's probably ways where, let me not start saying random stuff. But there's probably ways where you can improve the method. Yeah.
I just want to say it should be replicable. So
Yeah.
you should have the same result every time you do the same sequence.
Yeah. So one important thing that happens to me, that's actually a very valid point. So when I send for sequencing, sometimes the sequence comes back as N, and I don't know which one of the nucleotide it is. So I have to resequence if that piece is of interest, okay? And the reason for that, why it happens, is that it's going too fast. Like the sequencing is going so fast that sometimes the wavelength is not picked up properly or things like this. These machine errors need to be kind of decreased. What's your name? Maria. Sarah, Matthew, Julian, Arthur. Who else? That's it. This side doesn't want points. Okay, so that's it for today. Is anyone coming to the office? No? Okay, cool. So I can leave.
Yes, Simon, can we talk about politics at that time?
Me, I'm okay with it as long as there's the science too. Actually, I love politics, not because when I see things like this map stuff, it annoys me. So I have to talk about it, okay? Someone has to do it. My father is a professor. He told me don't do politics, but you know what? We're doing politics. But as long as you use the science behind it, because it's important to know that politics shape how we do science. And if I don't want to get too much to the side because I might cause problems to myself, but the way you do science is not based on what you see in the movies where there's this guy that's doing research to cure this or whatever. It's more like who's funding who knows who, and then who's getting this approved and that approved, right? For example, paper review. We know that my lab works on a specific protein doing these specific techniques. So the reviewers, they don't know it's our lab, but they see the research, they know it's us. So it's kind of blind, but not blind. And so there's favoritism happening in there, and if you know people, etc. And funding is the same thing when you're reviewing grants. If you see the project, and obviously, just like insider trading, where the company tells you, someone from the company tells you this company is about to do this move, so invest in it the night before, and then the stock goes up. This happens too in science, okay? So politics do shape the science, and this is why I like to talk about it. Okay, it's not to put myself in deep shit and then get fired, okay? But it's important. So yes, for sure, you can do that. Okay? Take care, guys. See you next week.